Okay, it's 9:01 a.m. and I see that people are still joining, but in the interest of time, I think we can start. Welcome everyone once again to the agroecology webinar series. My name is Evelyn Zuniga, and I am one of the communications officers for the Agroecology Partnership Project. What is the Agroecology Partnership? The partnership officially started in 2024 and is a project funded under the Horizon Europe program. It currently unites the European Commission and 72 partners across 26 member states, associated countries, and third countries. The Agroecology Partnership seeks to support the agriculture sector to meet the targets and challenges for climate change, biodiversity loss, food security and sovereignty, and the environment. The partnership combines the resources of the European Commission and participating countries to fund advanced research and generates essential knowledge and innovative technologies closely aligned with its primary themes and goals. These are second webinar in the series Agroecology Soils. And throughout this series, we will explore the key practices, challenges, and innovations that contribute to maintaining healthy soils for resilient and productive ecosystems. I just have a few key housekeeping announcements. Please feel free to type your questions in the Q&A and the chat box, which I have enabled for everyone. And I'm going to do my best to answer as many questions as I can with the time that we have, which is around 45 minutes. Today, we are very fortunate to have Dr. Alessandra Trinchera as our panelist. Dr. Trinchera is currently working as a researcher at the Council for Agricultural Research and Economics, CREA, in Italy, in Rome, Italy. Dr. Trinchera's research interests include circular economy for fertilizer production, crop diversification through rotations, intercropping, strip cropping, agroecological service crops, below ground functional diversity in cropping systems, focusing on soil microbial communities and beneficial symbiosis, nutrient cycling and soil organic matter storage, and phosphonic acid contamination of organic products. Dr. Trincheras has been involved in multiple national and transnational projects like Biophos Wine, Deer Farming, Surbeck, among many, many others. Dr. Trincheras, thank you very much for accepting our invitation to present your research today. Oh, sorry, I forgot to mention. The title of her talk today is Agroecological Strategies from an Efficient Functioning of Plant Soil Biota Interaction to Increase Carbon Sequestration, which is part of the AgroEcoSeqC project from EJP SOIL. As once again, Dr. Trinchera, thank you very much for joining us today. And I'm just going to stop sharing my screen and you can start sharing your presentation. Thank you so much.
Thank you so much, Evelyn. Thank you. I'm very happy to be here.
Thank you so much.
I share my screen.
Perfect. Yes.
Perfect. Okay. So I would eliminate this part because I have part of the screen. Okay. Perfect. So good morning to everybody. I'm very happy to be here and to have this chance to talk about the results of AgroEcoSeqC project. AgroEcoSeqC was a project funded by the European Commission within the EJP SOIL program, a program which collected a set of different research projects with the different topic, different objective. The AgroEcoSeqC project was based on defining the agroecological strategies for an efficient functioning of plant-soil biota interaction to increase the soil organic carbon sequestration. We know that in an agroecosystem, different biotic and abiotic factors work together in order to make the system more efficient in terms of nutrient use efficiency, carbon sequestration, and so on. It is very relevant to understand, to deep know the mechanism involved in this interaction between the different factors, because this knowledge allows us to manipulate the agroecosystem in order to optimize the production of the system, but also in order to save resources, lower the impact of the management practice in the field. In this last year, we know that we, in Europe, but not only in Europe, we are suffering of the effect of the climate changes that heavily affected the soil organic carbon sequestration that decreased over time, but also we observed a decrease of the soil biodiversity in our countries. So we need to face this emergency by considering the chance to engineering the agroecosystem. We have in our hands the possibility to select, for example, the different crops on the basis of their target traits, for example, by using a high diversity, crop diversity in field. We can manage the input we used, for example, by reducing mineral fertilization in favor of organic amendments. But at last but not least, it is important to stress the need to reduce the soil disturbance. And as you know, minimum tillage, no tillage are practices that are introduced in several countries in order to limit, to lower the carbon loss from the soil. All these potential approaches can help to increase the ecosystem services. I have listed some of them, for example, the promoting of soil aggregate stability, the sustainment of soil organic carbon accrual. We can improve the nutrients availability and their use. The increase of soil microbial biomass, but especially favoring the soil microbial richness and diversity. So work on the soil biodiversity as a whole. And then we can go in depth with the objective of the AgroEcoSeq project. So the AgroEcoSeq objective is the understanding of the effect of agroecological practices on soil organic carbon accrual, mediated by the plant traits and the soil biota functional diversity across different European pedoclimatic conditions. We studied different involved actors, starting from the plant species in field, then macro, meso, and microfauna, referring to the soil food web, the soil bacteria and fungi community, in particular diversity and functional diversity, and a focus on fungi-soil aggregate interactions. Of course, we do not have so much time to go in depth with all the results of the project, but I will try to give you an overview of the main results. This is the description of the European experimental sites that were evaluated in AgroEcoSeq project. We work in 7 long-term experiments and 2 new experiments that were chosen in Europe, ordering them under a European scale gradient of agroecological intensification. That means that we define a set of agroecological practices that were used together, 1 or 2 or 3 together, and the systems were ordered coming from the lowest level of agroecological intensification up to the upper one. That means, for example, an organic system, organic from more than 10 years, that allow to define a sort of conservative system from many years. But we can go from on organic orchards, agroforestry, a rotation with the mixed cover crop, and so on, up to the possibility to introduce, for example, some bioinoculant in the field. What is relevant is that in each these systems, we considered also a local scale gradient of agroecological intensification in each site. That means that in each EU experimental site, we considered 3 different levels of agroecological intensification. A lowest agroecological intensification, for example, by considering only the effect of soil disturbance where no tillage was introduced, and two highest agroecological intensification, where, for example, the absence of soil disturbance was coupled with the use of intercropping or cover crop or, for example, the crop residue restitution, etc. These two systems were compared to the control where no agroecological intensification was introduced. For example, monocropping without any kind of cover crop, etc. So we have T1, T2, and T3, different gradient of agroecological intensification in each site that were evaluated in a randomized block design with 4 blocks per each treatment in each of the experimental site in AgroEcoSeqC. Agroecological practice tested in AgroEcoSeqC project refer to reducing soil disturbance, as mentioned, highest plant diversity in field, for example, by using both cover crops or intercropping. We use Poaceae, Brassicaceae, Fabaceae species, or mixed species, agroforestry, organic systems, as mentioned. We go from monocrop to rotation or multicropping up to similar to grassland. We want to go toward a system that agro system that could be at the best similar to a natural system. It is an ideal, but we want to make this way in order to manipulate our system to be a sort of imitation of the natural ones. We use also beneficial fungi inoculants or organic fertilizer, compost, residues, crop residues restitution. This is a sort of list of the different sites considered in AgroEcoSeqC with the different treatment in order to have the idea of the number of the site and treatments considered. Here is the multifunctional approach from analytical point of view that we used. Starting from the soil parameters that are here reported, we also go in depth with the study of the plant community diversity in field in terms of by-species density and diversity indexes definition. But we go in depth with the functional diversity. That means identifying from the plant some above and below ground functional traits. For example, we refer to arbuscular arbuscular mycorrhizal trait, grass, percentage of grass plants, perennial trait, specific leaf area trait, and so on. We refer to macro and mesofauna functional diversity, considering ants, spiders, bacterivores, and fungivore nematodes, etc. And we go in depth also in depth with the bacteria and fungi community composition and functional diversity. Here we will refer in particular to Shannon and Chow diversity indexes and richness of the bacteria and fungi community. Then, at the end, we focus on the importance of the plant-fungi beneficial symbiosis, also taking into account their interaction with the soil aggregates. The final step was to modeling all this information in order to understand how the different soil biota can affect the soil organic carbon accrual. And let's start from the results. This is the first results that we observed. Here is the PCA, where we analyzed the set of selected indicators as affected by the site location, regardless the treatment applied in the field. The principal component analysis demonstrated that the variability of selected indicators is clearly explained by the experimental site. If you observe, for example, the variable, the indicators, soil organic carbon, you can see that in the right part of the graph, you find the Italian system, the French system, the Lithuanian system, that were those systems that we identified as highest agroecological intensification. While in the last part, we found, for example, the Spain and the Turkish, where we considered as less agroecological management intensification applied in the field. These can allow us to identify the role played by the site location when considering the effect on the parameters that we have, or indicators that we have considered in this project. But when we go in depth with the effect on selected indicators of the agroecological practices, regardless the site location, you can see that there is no explained variance in this figure. That means that we have to go in depth, considering not only the effect of the practices, but the effect of the practices in a specific site location. Because without this link, we cannot understand if the practices are effectively useful to improve the system in terms of biodiversity and in terms of soil organic carbon accrual. And this is what we found when we considered both the factors, so the European site and the practices applied in each site, regarding the selected indicators here reported. As you can see, we listed the 6 of these experimental sites because we need to reduce them. In terms of a first group of data, that means the soil organic pool, in particular the soil organic carbon and the soil water extractable carbon in water, and the water stable aggregates. In the second part of the graph are reported the PCR regarding the available nitrogen and phosphorus in soil and the bioindicators, in particular the bacteria richness and fungi richness, and the bacteria diversity and fungi diversity. And this is very clear that when we consider the T1 and T2 treatment, where the T treatment is the control, the red ellipsis is in the majority of the cases completely separated by the green ellipsis. It is not always found that the T2, that is the highest agroecological practices, is more effective in affecting the selected parameters and indicators. Sometimes it is the simple, for example, no tillage as T1 that it was able to well differentiate the systems. It is the case, for example, of Italian system that is an organic one, where the light green ellipsis was positioned in the right side of the graph, and corresponded also to the higher soil organic carbon content. That means that no tillage made a great contribution to this soil organic carbon accrual in this system. And in parallel, we can say that similarly in the Turkish system, we see that both the T1 and T2 were quite more overlapped, such as in the Belgian one. In these systems, we can say that the practices of crop residues restitution and the introduction of beneficial inoculant based on fungi, mycorrhizal fungi, make a high differences in terms of effect on soil organic carbon and on also on in particular biological indicators, and in particular on fungi bioindicators in Turkish systems. can go in depth with this system eventually when discussing. Now I want to go in depth with some specific aspect that we investigate in AgroEcoSeqC. In particular, we go in depth with specific plant traits correlated to the accumulation of soil organic carbon. In particular, we observed that we can use the canopy height, the specific leaf area, the percentage of grass-like species, the supporting arbuscular mycorrhizal trait, and the perennials, because these traits, plant traits, are related to the potential increased carbon sequestration in soil. These by biomass productivity and quality, increasing the carbon storage in soil aggregates, and increasing the carbon stock in mycorrhizal mycelium, fungi mycorrhizal mycelium. And these are the main relevant results that we observe. For example, in the agroforestry system, higher plant species richness and greater structural diversity was found in terms of plant structural diversity compared to the arable and vegetable system. So the tree crop coupled with cover crop, herbaceous cover crop, gave the best performance of the system. In vegetable systems, such as those of Turkey, where tomato was a crop, the cover crop and fungi inoculation favored the plant species with, in particular, specific leaf area. That means that this kind of bioinoculation, for example, favor the maximization of plant growth and soil carbon inputs in soil, mediated by this kind of plant specific zeolite rate. In the no-till system, we found the predominance of the same species, so the plant able to mycorrhize. So we found, in fact, greater mycorrhizal colonization in field and an improved soil structure. Now we can shift toward biotic factor. That means, for example, the microbiological component and the macrofauna and mesofauna component. When going depth with the fungi and bacteria community, here I have reported an interesting results obtained by the Spanish colleagues regarding the the grouping of the dominant taxa of bacteria and fungi community and the rare taxa of bacteria and fungi community. We observed in particular that several taxa appear as useful indicators of positive changes in the soil microbiota in different sites. And the fact that some specific rare taxa are so relevant as indicators give us the information that they can be used as benchmark for assessing the soil quality, and in particular, how the soil quality can be improved by specific agroecological management. This is an important result that we obtain in AgroEcoSeqC that is already submitted recently. But another interesting result was obtained in when going depth with what happens in the how can I say, at the below ground, because we observed that, for example, when no tillage in all the system where no tillage were introduced, we observed a higher mycorrhizal colonization intensity of the plant roots collected, sampled in the field, in the different sites. So this is a very robust result because it is a result that is not referred to a specific site, but to all the sites where these practices were applied. We think that it is important because no tillage reduced the disruption of the mycelium in the field. That will be also important on affecting the soil aggregates. But more, we observed also by the group of Sara Sanchez Moreno from Spanish that the total fungivore nematodes, that are nematodes who eat usually fungi and all the IFAL network produced by fungi, reduced the SOP. That means there is a correlation, a negative correlation between the total fungivore nematodes found in the sample with respect to the soil organic carbon contained. All these data are referred to the whole set of data of the European core sites participated to AgroEcoSeqC. And so this means that the presence of fungivore nematodes in some sites can affect the, in particular, the fungi community and thus the mycorrhizal fungi, and then decrease the soil organ, potentially decrease the formation of this mycorrhizal mycelial network. Why this is very important? Because the mycelial network act in particular on favoring the aggregation of the soil microparticles. For example, we have in literature found that the fine soil macroaggregates that correspond to 0.25 to 1 millimeter are generally dominated by the fungi. And we found that by crossing the data of mycorrhizal colonization in percentage with the soil aggregate diameter in millimeters, so the dimension, using the scanning electron microscopy, a new way, a new approach to evaluate this data, we observed a good positive correlation between mycorrhizal colonization and the dimension of the soil aggregate diameter. We found also a good correlation between the soil aggregate diameter measurement, the diameter as measured, and the amount of percentage of carbon contained in the soil aggregates. This measurement was performed by scanning electron microscopy coupled with electron dispersive analysis. And we found that, in particular, the highest average value of carbon correspond to the fine soil macroaggregates between 0.25 to 1 millimeters. In these samples, we found very frequently the presence of this mycelial network that you can see in this picture, that is a mapping of the carbon as element contained in the IFAL mycelium, here observed by scanning electron microscopy. We do not record a positive effect of a direct positive effect of mycorrhizal colonization intensity on the soil organic carbon in soil, but we stress the importance of the mycorrhizal mycelial network when we go in depth with the average aggregate diameter. That means that this mycelium is able to favor the aggregation of the soil particles and so limit both the effect, for example, potential of the erosion, but also the degradation, the fast degradation, the fast mineralization of carbon in the soil. Here I have the last analysis that we made that is related to the effect of the agroecological practices and the functional diversity on the soil organic carbon accrual by using a modeling approach. We use the partial least square structural equation models by the colleague Dylan Warren Raffa of the CREA that applied a plant functional trait to the plant functional trait, the fungal and bacterial functional group, the model in order to evaluate how the soil organic carbon is affected by this biotic component. We observed that no significant direct effect of the plant traits were found on soil organic carbon and water extractable organic carbon. But we observe also that the plant leaf area, the grass trait and perennial traits slightly affected the soil organic carbon. There is no significant effect, but a trend on affecting the soil organic carbon. Why? Because really it's not the plant that affect the soil carbon, because we know that in the dynamic of the carbon, it is the macrobiota, the main actor involved in. So it was observed that some specific group of bacteria, in particular fermentation and nitrogen and phosphorus cyanobacteria, and the arbuscular mycorrhizal fungi, such as the litter and dung saprotroph fungi, appears as most relevant and active groups that were involved in soil organic carbon degradation. What is what is the meaning? When we have microorganisms, bacteria and fungi that are able to degrade the litter and in general all the residues, the plant residues coming to the soil, the way of which these group that are very effective of soil organic carbon are able to fastly or lowly degrading these litter or other kind of organic material make the difference in conserving, in saving the soil organic carbon into the soil. And by summarizing, wrapping up the results, we can say that there is a strictly relationship interaction between the plant functional traits and the groups, selected groups in the field when degrading the organic material that come to the soil. In particular, the rare subcommunities are the main actors that could shift the system from a non-conservative to conservative systems, mediated by the agroecological practices that we have applied. The root mycorrhization is very important because it is a sort of a physical instrument, a physical tool by which the fungi community, and in particular the mycorrhizal fungi, can increase, as a sort of a glue, the aggregation of the soil particles by protecting the soil organic carbon and so increasing indirectly the soil organic carbon. We also have a sort of data that we want to give you. In particular, that no-tillage increased the root mycorrhization up to the 20%, the no-tillage increased the root mycorrhization up to 20%, which correspond to an increase of soil organic carbon over 10 years of Mediterranean system. That means that the soil organic carbon was increased up to 20% by the no-tillage mediated by the root mycorrhization, especially when this no-tillage is coupled with the organic amendments. The use of Poaceae and Fabaceae plants used as cover crop increase the carbon accrual in the tested sites more than other type of species. For example, Brassicacea plants have not the similar behavior. But we also stress the importance to tailor the agroecological management in order to address in a multifaceted context specific European environmental challenges, because the role of the experimental system and so the system in which we apply the agroecological practices are very important in order to identify the best agroecological practice to be introduced to obtain the real objective. So I would thank all my colleagues that contributed to the AgroEcoSeqC project that I have the honor to coordinate, and thank you for giving me the opportunity to think about these results with you, and I hope to have interested the audience from our results. And thank you so much.
Thank you so much, Dr. Trinchera. It's fascinating. It's very interesting project with so many results, and you have to work with so many variables. Like it's incredible, and also like in different parts of the world, different like soil types, etc. So in your the last part you mentioned about the mycorrhiza and how beneficial is for the soil and the carbon sequestration. So I was wondering, do you think or how feasible is like to create different formulations of mycorrhiza to inoculate the soil like in different like regions? I imagine there will be different formulations depending on the soil, depending on the region, and inoculating the soil to try to recover the soil before like planting again. What do you think?
This is a very interesting question because this is one of the frontiers of the new inputs, organic inputs to the soil, because we are strongly working on that. We have, first of all, observed that, of course, not all the mycorrhizal fungi are the same, because specific mycorrhizal fungi developed in a specific environment. So it is important to identify those mycorrhizal fungi that are in a specific site, and the potential of the industry of fertilizer is working on identifying those groups of mycorrhizal fungi and multiply to create specific inoculant, site-specific inoculant. This is one of the main topics that, in my opinion, should be addressed also at European level, because it is important to work on this kind of inoculation. Of course, there are already on the market, in European market, several products that could be usefully used and applied in the soil, but it is, in my opinion, possible to work in this way, to focus on a specific components that could be exploited in the site, because this is a real frontiers.
Yeah, for where I can see like it can be really tailored to a specific site and the
Absolutely.
soil, and yeah, that's very, very interesting. Okay, so we're having some questions here. I forgot to mention, I was so excited about my question. You can put your question in the chat section. I have enabled for everyone, and then in the Q&A we already have some questions. So it says the first one, how do you differentiate the data linked to soil type? The data linked to the soil type.
Yes, of course, I have not reported all the data that we collected from each site. You have to consider that each site, apart from the Turkish and the Danish one, were long-term experiment, European long-term experiment that were consolidated in the years. So we have at least 10 years of long-term experiment. All these systems were characterized very deeply. That means that we, first of all, collected all the data regarding the texture, the pH, the amount of soil, sand, clay, loam, in order to have an overview of the soil characteristic from a physical, chemical, and biochemical point of view. Otherwise, it's not impossible to. I do not go in depth with some specific information because we have 25 minutes theoretically, and it's very strictly. It's very difficult to give you some information, but we evaluated all the site, and in also considering the different plot that we have evaluated.
Yeah. Yeah, so I just want to say that the webinar is going to be recorded. So in your last slide, you showed all your papers, so people can go and refer to those papers with all the details.
Yes, these papers are the papers that are already published, that are not always related to specific results of AgroEcoSeqC, because of course the project is ended, as a matter of fact, on December 2024. So we are submitted a series of a set of new articles that are referred to what I have showed you, but are still now under processing. So you can have an overview of the approach used in AgroEcoSeqC from those I have reported in the presentation, but we hope in the last months we hope to have the publication of the submitted papers, so we can eventually send you some link, for example.
Yeah, and it's going to be in the website also from AgroEcoSeqC. So yeah, perfect. We have another question here. It says, thank you, great results. Likely the main driver for shifting the agriculture from non-conservative to conservative systems is driven by profitability for farmers. How was the plant performance or yield response in these experiments, and how yield was related to the different soil metrics?
Yes. In this project, of course, we have all the data of production because we considered, for example, all the biomass produced in the different systems, and we refer to the different level of agroecological intensification applied in each site. We can have the data regarding the production results with respect to the different applied practices. We have not discussed on that because the project was an EJP SOIL project that was dedicated to a specific objective that was requested in the call, that was verify how the practices was able to increase as a matter of fact, the CREA, the carbon use efficiency, and the ability of the system to saving carbon. But we have some data regarding the biomass produced, and we can say that sometimes there is, when applying the agroecological practices, an increase of biomass. Sometimes we have a little bit a decrease, but we have not a significant decrease of result, especially for example, in case of the organic system. The organic system have an advantage, for example, from this kind of approaches. The no-tillage in our system is better because it allows to save water, for example, saving water better, especially in Mediterranean system where the water is a key point. And we have an advantages by using cover crop that cover the soil, so maintain the water, and favor the tree crop in order to uptake water and nutrients from the soil.
Yeah, definitely. Yeah, that makes sense. I have a question here. So, what do you think will be kind of a challenge or the biggest challenges in the future, like to try to ensure that farmers can adopt the practices recommended by the project? And yeah, how would kind of in the future maybe the project or a future project address this?
The main challenge, in my opinion, is as correctly underlined by the participant that made the previous question, is the productivity, of course, because the farmers are surely aware of the importance of the sustainability of the practices and for saving their system on long term in terms of environmental sustainability. But in my opinion, the real challenges is to couple the agroecological practices to a good performance of the system in terms of productivity. It is not a case that we are working recently in a project, in a European project, is a call of agroecology that is asking for identifying strategies for saving nutrients, water, and carbon, but by using a co-innovation approach with the farmers. So we need to work with the farmers in living lab by working with them in order to identify the main problems that they have to face. For example, the problem of plant defense that is very relevant with climate change.
Yeah.
So plant defense is one most important critical point, but also saving water and nutrients.
Okay. Yeah. And I'm
So
just conscious of the time. I have one last question. Um, what have been the kind of the most surprising or challenges, like challenging findings that you found in your project?
the most important one, I am very happy to have demonstrated that, of course, not by only me, but all the group, because this was a coral group that works together a lot. But we succeeded in demonstrating the importance to look not at a specific indicators, but to identify a set of different actors involved in make the system functioning at the best. It's not possible to think that I put something in the soil, a specific new fertilizer, a specific plant defense product, or a specific practices, and obtained what I need. I have to think that the system is very complicated, very is built by a network of different relationships between the actors, and we have to move all the actors. And so we have to apply different strategies that should be tailored, that should be really painted on specific systems. I think that the last slide that I show you is perhaps the first time that different actors were put together, showing how they act in order to identify how to best manage the system, and especially soil organic carbon accrual and sequestration, because this is an important point. And we have a lot of other tests that we made in field. We work with the labeling, carbon labeling, nitrogen, etc., because we had a lot of data from AgroEcoSeqC, but it is impossible to show all the data that we obtained, of course. But I think that probably with the publication we can give you more and more information.
Yes, definitely. And yeah, we can really show all the data. It's super interesting, but like I'm already like I think 7 minutes past. It's so interesting. So I want to thank you so much for accepting your invitation. I'm getting messages like people are very happy and they learn a lot from your talk. Thank you everyone for your questions and your interactions. And if you're interested in more of the project, you just have to type AgroEcoSec in Google and then you'll find it's the first result. It's very interesting project. And thank you everyone for being here today. Thank you, Alessandra.
Thank
Thanks so
you.
much.
Thank you so much. Thank you
Okay,
for
bye
this
bye
chance.
everyone.
Bye bye. See you.
Bye.